Our next speaker is Shirit Einav from Stanford University, and she will be talking about host hepatitis C virus protein interactions.
Thanks for the organizers to giving me the opportunity to speak here today. It has been a very exciting day, and thanks for Chris Jones for setting up the stage for this talk, so I could really skip most of my background slides. I think now we are all aware that hepatitis C viral infection is a major global health problem, estimated to infect over 170 million people, for which current therapies remain inadequate. It is the #1 cause of hepatocellular carcinoma in this country, which is theoretically preventable cancer with more effective antivirals. It is also the #1 indication for liver transplantation in this country. And in the absence of effective vaccines, certain populations continue to be at high risk for new infections. This is a cartoon of the HCV genome. It is a positive single-stranded RNA that is translated into a single long polyprotein, which is then proteolytically cleaved into individual proteins. They are the structural proteins that form the mature virion, and the non-structural protein that at most part are involved in replicating the HCV RNA. Today's talk is going to focus on the core protein, or the capsid protein. Again, very briefly, as Chris has mentioned, interferon and ribavirin have been the standard of care regimens until very recently, when some direct-acting antivirals have been approved. And while response rates are somewhat improved, there are still substantial challenges that have to be overcome. And our approach to target HCV, rather than by targeting viral functions, focuses on targeting host partners, with the hope that the involvement of a host cell component will reduce emergence of viral resistance, will provide pangenotypic coverage, and also by targeting host partners that are conservatively required across viral families, this will provide means for broad-spectrum antivirals. And here are some, these are the viruses that we're currently focusing on the lab, in my lab, HCV, HIV, and dengue virus. And we're primarily focusing on using some novel technologies for screening for viral host protein-protein interactions on a large scale. And today I'm just going to focus on one of the stories we've been recently focusing on, which has to do with a novel viral host interaction related to HCV assembly. So HCV assembly requires a coordinated integration of cellular factors as well as viral factors that have to bring together the core, the capsid protein, with the two envelope proteins and the viral RNA. It remains the least understood stage of the HCV life cycle, and this is in part because it was only possible to study this stage after the recent establishment of a complete HCV replication system in cell culture, and it is currently not being targeted by approved drugs. The current understanding of HCV assembly suggests that viral particles, or the core protein, accumulates on the surface of lipid droplets, which is quite unique for HCV. Envelopment is then thought to occur on the surface of the ER, where the envelope proteins are retained, and this is where the virus becomes infectious. And then release or regress is thought to occur through the secretory pathway, where the virus is associated with VLDL structures. This is a schematics of the core protein. Its C-terminus region is being cleaved, which allows the mature form of the protein to then localize to the surface of lipid droplets. And very nicely shown by Melanie Ott's group, DGAT1, an enzyme essential for lipid droplet biogenesis, has been shown to bind core and to localize it to the assembly sites on lipid droplets. So the current model, again, suggests that core accumulates on the surface of lipid droplets, and then envelopment occurs here on the surface of the ER. But what are some of the viral host determinants that are participating in those later stages of viral assembly? And how does core traffic from the early assembly sites to development sites remain some important unanswered questions? Cellular membrane trafficking processes depend to a large extent on the function of tyrosine signals. These are in the form of YXXo motif, with Phi being a bulky hydrophobic residue. And such motifs reside within the cytoplasmic regions of membrane host cargo proteins and are known to be recognized by the mu subunit of clathrin adaptor protein complexes, or AP complexes. There are 4 such complexes that have been identified in mammalian cells, designated AP1 through 4, and these are known to be involved in distinct membranous trafficking processes. We've recently identified a heretofore unrecognized YXXo motif within the HCV core protein. Shown here, this motif conforms with the YXXo motif found in those cargo proteins I just mentioned to you, with some examples shown here. These proteins are known to bind the mu subunit of clathrin adapter 2, or the AP2M1 protein, which is an early endocytic protein. Here are some examples of retroviral proteins which harbor such motif and are known to bind AP2M1, and it was shown both for HIV and additional retroviruses that AP2M1 is in fact essential for infectious viral production. Here's the consensus sequence for all HCV isolates available in databases today, and as shown, this motif is very conserved across all HCV isolates within core. And we therefore hypothesized that core's YXXo motif may mediate binding to AP2M1, and that this interaction may be essential for mediating late stages of the HCV life cycle, and therefore approaches designed to disrupt this interaction may be used to inhibit infectious viral production. So our first aim was to first determine whether core indeed binds AP2M1 and the role of this motif in AP2M1 binding and infectious viral production. We then wished to study the role of AP2M1 in infectious HCV production, to then determine the mechanisms that regulate this interaction, and finally to try to translate these findings into the development of novel antiviral strategies. So starting with the first aim, we had a little challenge to overcome, since interactions between sorting signals and clathrin adapters or endocytic proteins are notoriously weak with KDs at micromolar range. They're transient and they involve membranous proteins. Therefore, very difficult to study by current technologies, and this is by no means specific to this interaction. Lots of the interactions we try to characterize fall within these categories, as most of the HCV proteins are membranous, just like other RNA viruses. So we were heavily involved in developing novel technologies to overcome these challenges, and one of these technologies relies on microfluidic affinity assays. Here's an example of a microfluidic chip aligned and bonded on a glass slide. It is composed of tiny reaction chambers that are connected with each other. These connections can be opened or interrupted by activation of micromechanical valves. And microfluidics, affinity assays have been originally developed by Dr. Stephen Quake at Bioengineering in Stanford, and he has originally used it to study interactions between proteins and DNA. As a postdoctoral fellow at Stanford, I've then developed an assay to look at interactions between proteins and RNA, and also to screen a large library of small molecules for inhibitors of such interaction. And more recently, we've developed an assay to look at viral host protein-protein interactions. This is a high-throughput screening assay. And the hallmark of this format is that it allows studying weak and trans interactions by allowing mechanical trapping of molecular interactions on the device. It also allows nanoliter protein consumption, which then enables one to express the proteins of interest in vitro using mammalian expression system. By adding microsomal membranes to these lysates, one can then look at membranous proteins. And there's some additional advantages that allow us to really use it in high throughput manner, which is less relevant to today's talk. So here's just some representative immunofluorescent images from the device. So in the green channel, you could see the expressed human protein, the AP2M1, on the surface of the chip. Here's our core protein, the bound viral prey, which seem to bind AP2M1. Here's a control protein, the non-structural 3 protein that did not bind. And the right panels represent the overlay of the green and the red channels, essentially allowing us determination of prey to bait ratio. And here's the binding curve of increase in concentration of the core protein on the control NS3 protein to AP2M1, suggesting that indeed core binds AP2M1. We complement this in vitro format with high throughput protein fragment complementation assays. This specific format relies on fusion construct of the prey and bait proteins to 2 halves of a Gaussian luciferase. These constructs are then delivered into cells, and cells are lysed and subjected to standard luciferase assays. This specific format was optimized by our collaborators at Pasteur Institute, and Gregory Neveu, who is a postdoc in my lab, has been involved in developing this format. It is highly sensitive and again allows studying membranous proteins in their natural cell model and natural compartment, and again has some additional advantages for high throughput screens. Using this format, we were able to show again that core binds AP2M1. In fact, it binds the two isoforms of AP2M1, A and B. Binding does not seem to be genotype specific, as core from two genotypes, both the 2A in black and 1B in white columns, demonstrated significant binding, unlike some negative controls here. The right band represents transferrin receptor, which is a host cargo protein harboring a YXXX Phi motif known to mediate binding to AP2M1. And as shown, the apparent affinity of AP2M1 to core is about twofold greater than to transferrin receptor. Binding appeared specific as when increase in concentration of either free AP2M1 or core effectively competed for core AP2M1 binding in the system, unlike an unrelated protein. As good virologists, we then wanted to demonstrate this interaction in the context of HCV infection, and while this was not easy, we managed to do so by using AGOH 7.5 cells that were infected with HCV. We prepared membrane fractions from these cells, and again, because of the transient and weak nature of this interaction, we then incubated these membranous fractions in the presence of a DSP linker to allow covalent binding of the already bound complexes, and then subjected the samples to immunoprecipitation and Western blotting. As shown here, anti-AP2M1 antibodies, but not IgG controls, were able to effectively pull down core. And when samples were incubated in the presence of caliculamin A, which is a compound that locks AP2M1 in its active bound conformation, dramatic increase in binding was observed. And we were able to demonstrate binding in reciprocal conditions as well. This is confocal microscopy images demonstrating that the AP2M1 in green and core in red significantly colocalized in the context of HCV-infected cells. To study our hypothesis that the YXXX Phi motif mediates binding of core to AP2M1, we introduced mutations, alanine mutations in both the Y and the Phi residue, as well as designed a mutant, a V2L mutation, which represented swapping of the 2A YXXX Phi motif with that of the 1B genotype. These mutations had no effect on core level of expression. Nevertheless, they had the alanine mutations had significant effect on core AP2M1 binding, and no such effect was demonstrated with the V2L mutation, suggesting that indeed this motif mediates binding to AP2M1. We then introduced these mutations in the context of the HCV genome, and in particular in luciferase reporter genome, which replicates and produces infectious virus at high titers. In vitro transcribed RNA of either wild type or mutant virus was electroporated into AGOH 7.5. These are human hepatoma cells. Cells were lysed and subjected to luciferase assays at 6 hours and 72 hours as a measurement of HCV RNA replication. Supernatants were harvested at 72 hours, were used to inoculate naive cells as a measurement of extracellular infectivity, again by luciferase assays. The same supernatants were subjected to measurement of HCV RNA as well as the core protein by either real-time PCR or ELISA, respectively, as measurement of non-infectious viral production. And cell lysates harvested at 72 hours were again used to inoculate naive cells as a measurement of intracellular infectivity. So using this panel of tests, we've demonstrated that the Y and Phi mutation, the YXXX Phi mutation, had no effect on HCV RNA replication, as both at 6 hours in the white columns and at 72 hours in the black columns. Nevertheless, these mutations had dramatic effect on both extracellular infectivity on the left and intracellular infectivity on the right. Please note that these are logarithmic scales, so there was about a 3-log reduction in infectious viral production induced by the Y mutation and an intermediate phenotype with a Phi mutant, whereas the V2L mutation behaved as wild-type virus. The Y mutation behaved essentially as an assembly defective mutant, which is deleted for the entire sequence of the 2 envelope proteins. And as I mentioned, the degree of the effect on infectivity correlated with the effect of these mutations on AP2M1 binding, thus providing a nice genetic validation of this target. Now, the effect that the extracellular infectivity phenotype mirrored that of the intracellular infectivity suggests that this motif is involved in mediating HCV assembly and not release. Since these mutations led to production of HCV RNA and core protein in the supernatants, which did not exceed that of the assembly defective mutant, we concluded that no infectious viral particles were formed as a result of these mutations. And importantly, after passaging the mutant virus over a few weeks, we were able to detect reversion of the infectivity phenotype, which correlated with the emergence of primary site reversion, thus providing further validation for the requirement of a functional YXXo motif for efficient infectious viral production. Then moving to our second aim, looking at the role of AP2M1 in infectious HCV production. So in order to study our hypothesis that the AP2M1 mediates HCV assembly, we first established stable clones harboring shRNAs that target various regions in the AP2M1 gene. And shown here on the left is effective depletion on the protein level, and on the right on the RNA level. These clones were then subjected to the same battery of tests that I've demonstrated earlier. And as shown here, AP2M1 depletion had no effect on HCV RNA replication, again, either at 9 hours or 72 hours. Nevertheless, just like the Y mutation, AP2M1 depletion dramatically decreased both extracellular infectivity and intracellular infectivity, and the degree of inhibition correlated with the degree of AP2M1 suppression, suggesting that AP2M1, again, is essential for HCV assembly. We were able to rescue the defective phenotype by ectopic expression of a wild-type shRNA-resistant AP2M1, thus essentially excluding any off-target effect of the shRNAs. So AP2M1 is an early acidic protein that is mostly known for its involvement in trafficking membrane proteins from the plasma membrane to the early endosome. So how is it possibly involved in mediating assembly of a virus that occurs on the surface of the lipid droplets and then targeted to assembly sites on the ER? So the first question we had is whether there's AP2M1 on lipid droplets in naive cells, and the answer is essentially no. We used quantitative immunoconfocal microscopy analysis for this, and essentially there was no significant localization of AP2M1 to lipid droplets. We then looked at infected cells, and unlike naive cells, there was significant localization of AP2M1 to lipid droplets in these cells, similar to in its extent to localization of core to lipid droplets, which was previously reported, suggesting that in the context of HCV infection, somehow AP2M1 gets recruited to the surface of lipid droplets. We then asked whether the core's YXXo motif is involved in this recruitment. And to study this hypothesis, we overexpressed AP2M1 M-cherry either by itself or co-expressed it in the presence of either wild type or the core Y2A mutant. And just like in naive cells, again, AP2M1 by itself did not localize to the surface of lipid droplets. Nevertheless, when it was co-expressed with a wild-type protein, there was significant recruitment of this protein to the lipid droplets, which was disrupted in the presence of the Y mutation, suggesting that perhaps this motif is indeed mediating recruitment of AP2M1 to lipid droplets. We then wish to determine whether disruption of core AP2M1 binding somehow affects the subcellular localization of core. And to start looking at it, we looked at core localization to lipid droplets. So again, in wild-type virus, just as was previously known, there was some degree of core localization to lipid droplets. Nevertheless, in the presence of the Y mutation, there was dramatic increase of core localization to lipid droplets. The same phenotype was observed in AP2M1 depleted cells. So again, dramatic localization of AP2M1 to the surface of lipid droplets in this context. Of core to lipid droplets in this context. In contrast, disruption of core AP2M1 binding, either induced by the Y mutation or by AP2M1 depletion, decreased core localization to the trans-Golgi network, which is the organelle through which HCV is thought to be aggressing through. And last, disruption of core AP2M1 binding, in this case by the Y mutation, also dramatically decreased co-localization of core with E2, which is retained on the surface of the ER membrane. So taken together, this quantitative confocal microscopy immunofluorescence analysis suggested that the defect we see in assembly is probably following accumulation of core on the lipid droplets, but prior to the development on the ER membrane. We then wish to look at the regulatory mechanisms of this interaction. A specific threonine residue, threonine 156 in AP2M1, is known to undergo phosphorylation by two host cell kinases, AAK1 and GAK. And this phosphorylation is known to stimulate binding of AP2M1 to the host cargo proteins. And it's transient due to the activity of the PP2A phosphatase. Now, I previously shown you that caliculin A, which inhibits PP2A and thus locks AP2M1 in its activated bound conformation, significantly increased binding of core to AP2M1, suggesting that perhaps these regulatory mechanisms are involved in regulating core AP2M1 binding. So we then hypothesized that perhaps the threonine 156 to alanine mutations within the AP2M1 gene may decrease core binding and infectious viral production, and also that perhaps AAK1 and GAK may be regulating this binding and are essential for HCV assembly. So to start studying this hypothesis, we first introduced the T156A mutation into our AP2M1 sequence and study the effect of these mutations on core binding. And indeed, using either the PCAs format or the microfluidics format, we showed a significant inhibition of core binding. When we overexpressed AP2M1 harboring these mutations, we demonstrated no effect on HCV RNA replication. Nevertheless, these mutations, the overexpression of this mutant significantly decreased extracellular infectivity in black and intracellular infectivity in white, suggesting that this mutation essentially had a dominant negative effect on infectious viral production. Excuse me. We then moved on to AAK1 and GAK, and we depleted these genes by transient RNAi technology. This is the top panels demonstrate depletion on the RNA level, on the bottom on the protein level. And depletion of either AAK1 or GAK dramatically decreased core AP2M1 binding, suggesting that these may be involved in regulating this interaction. It had no effect on HCV RNA replication, but again, significant effect on extracellular and intracellular infectivity, suggesting an effect for these kinases on assembly. Last, we wish to try to translate this knowledge into novel strategies to combat HCV. And we looked at affinity binding assays of known kinase inhibitors and identified several compounds which bind either AAK1 or GAK with very high affinities at nanomolar concentrations. Now remember, AAK1 and GAK are not the known cancer target of these compounds, and they've not been studied in any viral infection, but they were looked at when some of these kinase inhibitors have been analyzed. And again, binding is with very good affinities. So we hypothesized that these compounds may, in fact, inhibit core AP2M1 binding and HCV assembly. And this is precisely what we saw. Here's a dose-response effect of increasing concentration of 3 of these compounds on core AP2M1 binding with IC50 in the range of 14 nanomolar. When we studied the effect of these compounds against infectivity, we showed significant effect on extracellular infectivity as well as intracellular infectivities, with EC50s in the range of 0.15 to 1.8 micromolar. Importantly, there was no effect of these compounds on cellular viability, as was determined by Alamar blue-based assays on the left. And surprisingly, there was no effect on HCV RNA replication, which is the stage of the viral life cycle that multiple protein kinases in fact are involved in, suggesting a reasonable selectivity. And as predicted, these compounds significantly inhibited AP2M1 phosphorylation. This is just their overall antiviral activity on HCV infectious infectivity with the KDs that are noted here. Now, these results essentially provide pharmacological validation for the importance of AP2M1, AAK1, and GAK for efficient HCV assembly. For those of you in the HCV field, there was a recent report about erlotinib's effect on HCV entry via its effect on EGFR. So we provide another mechanism of action for erlotinib, which is entirely independent of its effect on entry. And some of these drugs are already approved drugs for cancer, and there's an exciting opportunity for potentially repurposing these as antivirals. So in conclusions, core harbors a YXXo motif, which mediates AP2M1 binding and HCV assembly. AP2M1 is essential for HCV assembly. Core seems to be hijacking AP2M1 and recruiting it to the assembly sites on lipid droplets. Core AP2M1 binding affects the subcellular localization of core. AAK1 and GAK regulate core AP2M1 binding and are essential for HCV assembly. And last, kinase inhibitor with activity against either AAK1 and GAK are potent inhibitors of core AP2M1 binding and HCV assembly. Just some of the ongoing work, some of the unanswered questions we're currently trying to answer is whether this interaction truly affects trafficking of core from the assembly sites to envelopment sites. And I can tell you briefly that we've identified a similar motif in the E1 protein, the envelope protein, and our current models suggest that perhaps by binding both core and E1, AP2M1 functions sort of as a bridge that helps traffic particle from the lipid droplets to the envelopment sites. We have some evidence that core and E1 bind additional mutations of other clathrin adapters, and since each of these are involved in mediating other trafficking pathways intracellularly, we already have some preliminary data suggest that in fact these are involved in additional stages of the viral life cycle. And if this is true, then this has potential implications for treatment, since these inhibitors may then be in fact inhibiting multiple stages of the viral life cycle, which is of course relevant for emergence of viral resistance, potential for synergy with other antivirals and so on. This is relevant, seems to be relevant to other flaviviridae, such as dengue, where we identify similar motifs within structural proteins. And importantly, as I started by saying, HIV has been known to be hijacking AP2M1. What was not looked at before was whether GAK and AAK1 are involved in mediating HIV infection. And I can tell you that with in collaboration with the Katzenstein lab at Stanford, we already have some preliminary data suggesting that indeed depletion of AAK1 and GAK significantly disrupt HIV infection, and that the inhibitors have effect on HIV infection. So in summary, this is the first sorting signal mediating binding of clathrin adapters identifying the flaviviridae family. First evidence for involvement of AAK1 and GAK in any viral infection. These results provide novel targets for antiviral treatment directed at assembly, and inhibitors of AAK1 and GAK. Some are already approved drugs for cancer with opportunity for repurposing. These represent overlapping host cell partners essential for HCV and HIV, and possibly additional flaviviridae, and compounds with antiviral activity against HCV and HIV, and again, for which we're very excited to further determine the clinical utility. And I'd just like to thank our collaborator, the Pasteur Institute, David Katzenstein, my collaborator in Israel, who's helped with establishing the microfluidic format. And most importantly, the people who've done the lab were all in the audience: Gregory Navu and Rina Baruch, who have spearheaded all the work in this project, and Amod Zivav, who's helped with the microfluidic experiment. And thank you.
I have a question. Do you know already whether AAK1 and GAK, do they also locate in infected cells to lipid droplets?
We have not looked. It's a great question. It's something we should be doing. We haven't had a chance to do it.
I don't know. I just missed your question. I don't know if it's the same one, but I was very interested in the phenotype of the Y136A mutant, and that it accumulates on the lipid droplets. So can you allude a little bit more about on this? So you think that the AP2M1 is basically taking off the core from the lipid droplets and sort of guiding it into the ER sites where they're thought to be involved in, encapsulation?
So the question is, why does the core Y mutant accumulate on the surface of lipid droplets, and whether we hypothesize that AP2M1 is involved in trafficking? So we have not proven the mechanism of the involvement of AP2M1. I mean, we did show an effect on subcellular localization of core. What we are now trying to show whether it's truly effect on trafficking. We certainly have a lot of preliminary data to support that, but we are now collaborating with Brett Lindenbach, trying to use viral particles in live cell imaging to actually show core trafficking with AP2M1. We have some dominant negative results that support that that's the fact, but
and then you had another observation, which I might have missed, is that the knockdown of the AP2M1 is actually causing core to accumulate in the
The
transcon
same
network?
phenotype. That was we showed.
No, also enhanced lipid droplet.
Right, the same phenotype. So stuck on the surface of lipid droplets and depleted on the surface in TGN, or decreased localization with E2 on the ER. So
decreased in
right.
the ER. Okay, good. Thank you.
So it seems like it's a later stage than what you've shown with the DGAT1, which accumulated on the ER, right, prior to recruitment to the lipid droplet. So, yeah. All right. Thank you.
Thank you.